for future events. So with no further ado, I will turn it over to Dr. Zatloukal. Thank you.
Thank you for your introduction. In my presentation, actually, I want to highlight two topics. First of all, making the case why is standardization of sample pre-analytics so important? And furthermore, is what now are the specific pre-analytical requirements for metabolomic analysis, and I will highlight this with some examples from the research programs we have been involved in. The importance of reproducibility of scientific data has recently been highlighted in a cover story by The Economist. For instance, they found that many of landmark studies cannot be used by other laboratories. For instance, highlighted by a report from Amgen that they found that only 6 out of 53 landmark studies can be reproduced. So quality is an issue, and the question is how we can reduce the variations in scientific data. Big problem here is that even most advanced analytical technologies cannot produce better data than the quality of the biological material. So we have to take care the whole workflow from the patient, from the operations theater, from the clinics, to the analytical pipeline, that the biologically relevant imprint is maintained. And that's the same whether we talk about basic research, biomarker research and development, or drug development and target discovery. There is important knowledge developed in the environment of biobanks how to ensure the proper pre-analytical processing of samples for molecular analysis. These have been particularly addressed by two major programs, one under the umbrella of the National Cancer Institute in the United States, another one funded by the 7th Framework Program of the European Union, the SPEEDIA program, and these two initiatives try to do systematic investigation how the various steps in the pre-analytical process impact on various analytes in biological specimens. The challenge that these programs are facing is that there are several steps from the patient to the analytical procedure, and these slides highlight some of the steps critical for the processing of tissues. Problem is that some of these steps can be well standardized, but others not, because they depend on the specific requirements of the patient. For instance, you cannot control duration of surgery impacting on ischemia, medication given during surgery. So we all have to accept there will be some variation. This means when we cannot avoid variation, we have to know exactly what the impact on our analytical result is of these variable factors. And this becomes particularly important when we talk about the metabolome. On the one hand, the metabolome is so highly informative on the health or disease status of an individual because it integrates both environmental and genetic factors. But this at the same time makes the metabolome highly responsive to environmental factors and factors of the pre-analytical process. In order to investigate these parameters, we have performed studies during surgery in patients, this is liver surgery, in the context of a clinical trial protocol, where we could collect samples from the start of an operation and then could follow in the same patient how biospecimens respond during the various procedures and steps. In doing an NMR metabolomics profiling, one can immediately recognize that a sample responds to ischemia by increasing lactate and influencing glutathione concentrations. Now, having a more comprehensive summary on these changes in the principal component analysis of several metabolite profiles, and these are data from different individuals. All these lines correspond to the sample collected at different time points from a given individual, that you see an immediate change in this metabolic profile already after 30 minutes. This means you have to very well standardize exactly the time point when you collect the sample. Otherwise, you can hardly compare results from samples having different ischemia types. The next question was, how does the lifestyle influence the metabolome? And to answer this, we have performed a study in healthy volunteers. And actually, we monitored the variations of the individual metabolome over a 10-day study. There were 7 days, a regular lifestyle, well documented on all key parameters, and we collected during the day 6 times saliva, all the urine, stool, and blood. And then there were 2 days of lifestyle with a highly standardized diet. On the last day, highly standardized diet and standardized physical activity. So we wanted to see how strong does lifestyle impact on the metabolome of an individual. And the next slide summarizes these results obtained by NMR spectroscopy in a 2-dimensional principal component analysis. And these different colors highlight the metabolic signature of different individuals. So what you can immediately recognize that each individual has its highly personalized metabolome, like the genome. But also you see there is some significant variation in the samples collected at different time points. Having a closer look into these variations, like here, the samples collected from one individual during the different days of the study, visualized in the different colors, you can see there is a big variation from day to day, and even standardized diet does not really avoid this variation. And of course, physical activity had a significant impact. So what's the conclusion we can draw from this observation is, if you cannot really standardize lifestyle, which is not easy in a healthcare setting, you have to take multiple measurements. Then you can normalize for this, and then you get highly specific metabolomic profiles for individuals. Next question was, how stable is a metabolome upon storage? And so we did a pretty demanding setup together with Fraunhofer, IBMT in Germany to simulate various storage conditions. And what we found is, by this simulation, that it's not so much the storage temperature, it's the retrieval process that has an impact. And we have simulated storage and retrieval is -80 freezers, storage in liquid nitrogen gas phase, liquid nitrogen liquid phase, and a system that has also a temperature-controlled retrieval environment. And we found that free proline is a very good surrogate marker to monitor changes. And you can see that even liquid nitrogen, there is a significant increase in free proline upon repeated storage and retrieval. And actually, we found only a modest variation by storage in liquid nitrogen gas phase. Again, what we can learn from this is, it's not the storage temperature, it's the retrieval process that determines stability and robustness of metabolomic signatures in samples. And at the end of my presentation, I want to acknowledge all the co-workers and colleagues that contributed to this data and work, and last but not least, the funders who enabled this work. It was funded by the